Welcome to Educom. This is Imam with you. In earlier sections, we finished up to macromolecules. Now we'll start with the proteins. Proteins are polypeptides. Okay? It is a linear chain of amino acid linked by peptide bonds. Here, amino acids are linked by peptide bonds. Dietary proteins are source of essential amino acids. Dietary proteins means like proteins we consume on daily basis. So they are the source of essential amino acids. Amino acids are two types. We have essential and non-essential. Essential means we need such amino acids for our healthy growth. Protein is a heteropolymer. On the right side, the important the point to remember here, collagen is the abundant protein in animal world. Remember this, okay? Collagen is the most abundant protein in the animal world, whereas ribulose bisphosphate carboxylate oxygen is most abundant protein in the whole biosphere. Okay? Ribulose bisphosphate carboxylate oxygen is the most abundant protein in the whole biosphere, whereas collagen is the most abundant protein in the animal world only. Remember this. This is the most important in this page. Okay, and we have the structures of protein. So we have here the primary structures here. Okay, which are made up of polypeptides, means amino acids linked together with the help of peptide bonds, and we have secondary structures, which is of two types: alpha helix and beta plated sheet. Where we learn about this alpha helix and beta plated sheet during introduction to the cell, the unit of life. We have a scientist Linus Pauling. He is associated with the alpha and beta sheet discovery. Okay, and who was influenced by Linus Pauling? It is G. N. Ramachandran. Okay, who discovered the triple helical structure of collagen. Okay, so we can connect like that. So we have primary protein structure here, a secondary, okay, which is spherical, and we have a beta plated sheets, and tertiary, where we have a hydrogen disulfide. Okay, here we have, okay, and then we have a quaternary. Coming to this table, some proteins and their functions. Okay, collagen, intercellular ground substance. Okay, trypsin, that is a enzyme. So insulin is a hormone. Okay, these are all means the protein which functions at different places in different, their functions are also different. Antibody, that is also a protein but fights infectious agents. And receptor, sensory reception like smell, taste, hormones, etc., like how we smell, how we taste, and hormonal everything. And GLUT4, that enables glucose transport in cell. Okay, now from this we can also expect questions. Okay, we cannot ignore this table. Okay, sometimes questions are coming from the type, directly from the table itself. So trypsin, what is trypsin? It is amino acid, carbohydrate, like that it will come. So we need to thoroughly practice these things. So we need to understand the table, and also we need to understand the proteins and also their functions. So here GLUT4, which of the following protein is involved in glucose transport into cells? Okay, GLUT4, it is involved, it enables glucose transport. Okay, it enables glucose transport into cells. And we have antibody, and trypsin also, this is a specific word we have here. Okay, trypsin is an enzyme, and collagen, it is an intercellular ground substance. So what we understand from these different proteins and their different functions. Okay, coming to next slide, we have polysaccharides. Polysaccharides are long chain of sugars. Okay, long chain of sugars. So we have here sugar. This is a monomer. Okay, together they are forming disaccharide. The cellulose is a polymer of polysaccharide consisting only one type of monosaccharide, that is glucose. So cellulose consists of only glucose. So number of glucose molecules, they attach together forming a polymer of polysaccharide, that is cellulose. Cellulose is homopolymer, means heteropolymer means different, and homopolymer is same glucose, that is same monosaccharide, like that is glucose. So number of if you find if you see in the polymer in the chain, you will find only in glucose. For example, if it is a heteropolymer, then you have different sugars attached. Okay, in polysaccharide chain, the right end is called, this one is called reducing end, and also left one is called non-reducing end. Okay, and here inulin is a polymer of fructose. So like we have a cellulose which is a polymer of only glucose, which is a homopolymer. Inulin is a polymer of fructose. Plant cell wall is made up of cellulose, and examples you can take for homopolymer. Plant cell wall is made up of cellulose. Exoskeleton of arthropods. Okay, arthropods means like cockroaches, etc. Have a complex polysaccharide called chitin. Chitin is present. Chitin is a polysaccharide. Okay, it is where it is present. It is present in the exoskeleton of arthropods. They also have a building block, amino sugars and chemically modified sugar. They also contain the they have building block like amino sugars. The amino sugar means amino acid with sugar attached. Okay, and chemically modified sugars, which is you can take example like glucosamine, N-acetyl galactosamine. Okay, so here polysaccharides are long chain of sugars. Okay, they have a reducing and non-reducing ends, and cellulose is a polymer of polysaccharide consisting of only one type of monosaccharide, that is glucose, and we have inulin is a polymer of fructose. Plant cell wall which is made up of cellulose, and also chitin is present in exoskeleton of arthropods, and some polysaccharide they also contain sugars. Examples you can take glucosamine and acetyl galactosamine. Coming to nucleic acids. Nucleic acids are polynucleotides. Together with polysaccharides and polypeptides, these comprises true macromolecular fraction of any living tissues. Okay, we have proteins, amino acids, okay, nucleic acid, different. So among these nucleic acid, they represent the true macromolecular fraction of any living tissue or a cell. A nucleotide has three distinct components. One is heterocyclic compound, second is monosaccharide, and third is phosphoric acid as the phosphate. So a nucleotide should contain all these three compounds. You can see the example below, three parts of the nucleotide. Here we have a phosphate group, and here sugar, okay, and we have a base here, okay, that is adenine, okay, or purine bases such, and we have different examples for the bases. So we have pyrimidine, cytosine, uracil, which is found in RNA, thymine T found in DNA, purine, adenine, and guanine. AGCT, these are present in the DNA. The adenine, guanine, cytosine, and thymine, they are found in DNA. Okay, so they are polynucleotides, which together made up of a sugar, phosphate group, and also a nitrogen base. Heterocyclic compounds in nucleic acids are nitrogen bases named adenine, guanine, uracil, cytosine, and thymine. So just now we discussed about them in the above image. Coming to structure of proteins. Proteins are heteropolymers containing strings of amino acids. In organic chemistry, the structure refers to the molecular formula. For example, we have NaCl, MgCl2. This is the formula for sodium chloride and magnesium chloride. In physics, the physicists look up to the three-dimensional view of molecular structure. When it comes to biology, the structure is defined as food. Okay, in physics, three-dimensional view of molecular structures, whereas in biology, we define at 4 levels. So that we discuss in the image. The sequence of amino acids is linear array, that is from first amino acid to second, and the last is called a primary structures. Here we have a chain-like structure of the protein. So we have a single 1, 2, 3, 4 like number of amino acids, they join together, okay, and making a primary protein structure. Okay, primary protein structure means the sequence of a chain of amino acids. Okay, this is a primary protein structure. In secondary, hydrogen bonding of the peptide backbone causes the amino acids to fold into repeating pattern. Okay, so here, for example, it is in folded form like a staircase. Okay, so here what happened? Hydrogen bonding of the peptide. So peptide bonds are present in between the amino acid. So these are the peptide bonds. So again, hydrogen bonding of the peptide backbone causes the amino acids to fold. So by having another bonds here in the peptide bonds, okay, the protein folds into repeating pattern. Okay, causing the secondary protein structures. Coming to tertiary protein structure, three-dimensional folding pattern of protein due to side chain interaction. Okay, here we have seen the primary structure, how it occurs. It is made up of amino acids, okay, bonded together with the help of peptide bonds. Whereas in secondary protein structure, that is alpha helix and pleated beta plate shape, here we have a hydrogen bonding of the peptide backbone, okay, which causes the amino acids to fold into repeating pattern. Whereas in tertiary structure, we have three-dimensional folding. Like we have only in secondary structure, it only maybe it folds into 1 or 2 times. Here, tertiary protein structure, we have a three-dimensional folding pattern of a protein due to side interaction. So here, these folding, for example, it is from here it is folded like this. Okay, there will be interaction in this place. Okay, this is called the side to side chain interactions. Coming to quaternary protein structures, protein consisting of more than one amino acid chain. Okay, so proteins consisting of more than one amino acid chain. Okay, here different, like we have tertiary, we saw about tertiary protein structure, secondary protein structure, and also primary protein structure. Coming to quaternary structure, proteins consisting of more than one amino acid chain. So say for example, here we have one chain. Okay, here maybe multiple chains together, they intermingle and forming same, like side to side bonding in the chains. And a protein is imagined as line, the left end represented by the first amino acid and the right end represented by last amino acids. The first amino acid is also called as N-terminal amino acid, and the last amino acid is called C-terminal amino acid. Protein chain also fold in the form of helix. Some portions will be thread-like and some portions will be staircase. And also protein forms only right-hand helices. Okay, this is also important point we should remember. Okay, for the NEET it is most important. Protein forms only right-hand helices. Sometimes protein folds into other forms such as secondary and further folding gives tertiary structure, which gives us three-dimensional view of protein. Just now we discussed about this. Okay, here we have a linear chain of amino acid. We have a helical structure here, and then we have a three-dimensional structure here where the side-to-side interaction occurs, and same in quaternary protein structure where the different, like for example, this is one amino acid chain, so we'll be having such chains maybe more than one chain. Okay, some proteins contains more than one polypeptide or subunits, creating a tertiary structure of proteins. And here adult hemoglobin consisting of 4 subunits, 2 alpha, 2 beta, all together they constitute the human hemoglobin. Okay, this is also adult hemoglobin consists of 4 subunits, means protein, 2 alpha and 2 beta. So here you can take the example of this. Okay, 2 alpha and 2 beta, all together they constitute the human hemoglobin. Coming to the nature of bond linking monomers in a polymer. Okay, we have here one amino acid 1, amino acid 2. Okay, then we have a R group. Okay, we know that every amino acid it contains, okay, hydrogen, amino and variable R group. Okay, then the peptide, how the bonding occurs. Here what happened? So here the amine group. Okay, nitrogen is here. Okay, nitrogen is this is involved here. This area is involved. Okay, and making a peptide bonds between 2 amino acids. So what is released from this? Water is released. Okay. In polypeptide amino acids, all they are linked by peptide bonds which is formed between carboxyl group, amino group with the elimination of water molecule. Okay, you understand here, you can see clearly in the image 2 amino acids how they are joining together and how the water coming out of that reaction. Here we can see the structure of DNA, how exactly we talk about the structure. We have a sugar, we have a base, and we have phosphate. Same here you can see. Okay, it is two chains, they are coming in opposite direction. So we have a guanine and cytosine. The bonds are, we have 3 bonds, whereas thymine and adenine we have 2 bonds. You can see that in nucleic acid, phosphate moiety links with the third carbon of one of the sugar. Okay, this is the third carbon. Okay, this phosphate group is linking with the third carbon of one of the sugar of one nucleotide to the, okay, to the fifth carbon of the sugar of the succeeding nucleotide. Okay, here we have, this is one, okay, and this is here. Again, the bond between the phosphate and sugar is ester bond. Okay, this is the ester bond. Two strands of polynucleotide are anti-parallel. Okay, this is going this side and this is coming what this side. So now you understand, here nucleic acid phosphate moiety links with the third carbon of one of the sugar of the one nucleotide to the 5th carbon. This is the 5th carbon, this is the, okay, this is the connection happening between the 2 sugar molecules here. The bond between the phosphate and sugar is ester bond. This is the ester bond that I already written here, and they run anti-parallel. Those 2 strands, they run, the 2 strands of polynucleotide are anti-parallel. They run towards each other. Metabolism. Metabolism is a chemical reaction that changes food into energy. Then how we define metabolism here? It involves both catabolic reactions and anabolic reaction. What is catabolism? What is catabolic reaction? For example, here, this is the example for the catabolic. Here, in catabolism, the materials are break down and energy is released. Okay, whereas in anabolism, the smaller molecules they join together forming a bigger molecules consuming the energy. Okay, that is the 2 difference. Catabolism, energy is released. Okay, whereas anabolic reaction, they consume energy, means they anabolic reactions are energy dependent reactions. Metabolic pathways can be linear or circular pathways. Okay, they can be linear like a straight, sometimes circular. So here like this, from here the reaction occurs, then it goes through a sequence of events, coming again freely again join into the another reaction. So this is called a cyclic pathway, and this is a linear path. So linear pathways, so it will be released into the system. every metabolic reaction is catalyzed. So every metabolic reaction is the catalyzed. So you may be asked in exam like which of the following reaction is not metabolic, which is not catalyzed by enzyme. So remember every metabolic reaction is catalyzed. So every reaction needs enzymes. There is no uncatalyzed metabolic conversion in living system. Okay, there is no uncatalyzed, without involvement of enzyme. There is no any kind of metabolic activity in living system. Okay, that is 100%. Okay, that log that and remember these points here. Catalyst, the catalyst which fasten the rate of given metabolic conversion or also protein. The catalyst means, okay, like enzymes which are involved in the reaction. The catalyst which fasten means which increase the rate of reaction, which fasten the rate of given metabolic conversion are also protein. So before we learn that of hormones, enzymes, they are also protein. These proteins are called enzymes. Anabolic pathway leads to formation of more complex structures and consume energy. Here it is, you can see here energy is consumed, whereas here energy is released. The catabolic pathways lead to breakdown of larger and complex structure into smaller units or smaller molecules, and which also releases energy. And living organisms trap and store energy released during the reactions in the form of bonds. Okay, how the energy are stored in the living system? In the form of bonds, and when required, bond energy is utilized for biosynthetic, osmotic, mechanical work that we perform. Okay, so living organism trap and store the energy released during the reaction in the form of bonds. Okay, and when required, bond energy is utilized for biosynthetic, osmotic, and mechanical work that we perform on daily basis. So here, let me erase this. We have a amino acids which forms protein and their structures. Fatty acids we have, they form phospholipids which are main constituent of the membranes. Okay, and we have carbohydrates. Carbohydrates, they have monosaccharides and also glycerol. Glycerol can form the phospholipids, they can join with the fatty acids. Where monosaccharides, lot of monosaccharides joining together forming polysaccharides. And nucleobases, that is nucleotide, DNA and RNA. So in this section, are we clear? So metabolism means it's a chemical reaction that changes the food into energy. Like we are for example, here we have two examples for metabolism include catabolism and also anabolism. Where in catabolism, the energy is released into the stored. You can say in store. Whereas in anabolism, the stored energy is consumed for the reaction. Okay, thank you.